Hello viewers, I am Saurabh Mukherjee. Today I am discuss an important topic about photosynthesis, that is bioenergetics of photosynthesis, which generates the reducing power. Before starting the video, I am requesting you kindly subscribe my channel and press the bell icon for quicker notification. So let's start the video. First of all, what is photosynthesis? Photosynthesis is a process where light energy is transformed into the chemical energy with the help of light, CO2 and water, whereas oxygen is produced as a byproduct. Next is sites of photosynthesis, where photosynthesis takes place. Photosynthesis mainly takes place in chloroplast of the mesophyll cell. So this is a chloroplast which is double membrane structure. The chloroplast contain a granular stroma, which is a fluid-filled matrix-like structure, and another one is thylakoid, which is a flattened sac-like structure where light reaction of the photosynthesis takes place. Another important things chloroplast contain a circular DNA, which is known as plastic DNA or chloroplast DNA. The genome size of the chloroplast DNA is 120 to 217 kilobyte. There are two phases of photosynthesis. Number one is light dependent phase, where the light is must required, and number two is dark phase or dark reaction. Dark phase does not mean the reaction occur in the dark condition. It means there are no requirement of light. Light reaction occurred in the thylakoid of the chloroplast, whereas dark reaction occur in the stroma of the chloroplast. Here CO2 is fixed
and produced ATP and NADPH, which was further utilized in the dark reaction.
Now comes to the photosynthetic pigment. The photosynthetic pigment is the chlorophyll, which is the only light absorbing colored molecule. Chlorophyll is present in the thylakoid membrane. It is a 5 heterocyclic ring shaped system surrounded by a magnesium ion. A phytol ring was present or attached to the ring D. There are two major types of chlorophyll, that is chlorophyll A and chlorophyll B. The R group present in the B ring differs the chlorophyll A and chlorophyll B. The other pigment, which was also called the accessory pigments, are extend the range of light absorption. For example, carotene, xanthophyll, phycobilins are the example of accessory pigment. This chlorophyll funnels the absorbed energy to the reaction center. Basically, the chlorophyll which was attached in the thylakoid membrane are associated with accessory pigment. Absorption of photons excites this accessory pigment, which funnel the energy transferred into the reaction center. This reaction center photoexcitation results in a charge separation that produces a strong electron donor and acceptor. In plants, two reaction centers works as a tandem. First of all, number one is Photosystem II, which is a pheophytin quinone type of system designated as P680. The oxygen evolving complex attached to this system, here splitting of the H2O molecule occurs. Excitation of its reaction centers drives electron through the cytochrome B6H complex with concomitant movement of proton across the thylakoid membrane. And number two is Photosystem I, which was designated as P700 reaction center. From PS2, that is Photosystem II, the electrons are transferred to the Photosystem I, resulting the excited Photosystem I passes electrons to the FES protein, that is ferridoxine, then to produce NADPH. So this is the overall pathway of the electron flow between two photosystems. This is the Photosystem II and this is the Photosystem I. Here water was splitted by oxygen evolving complex, and electron is transferred from water molecule to the P680, and electron excites the Photosystem II, and electron transferred to a series of chlorophyll molecule, resulting this electron transferred to the cytochrome B6F complex, and then plastocyanin, which is only copper containing carrier. From the plastocyanin, the electron is transferred to the Photosystem I. After that, electron is transferred to the A0, A1 and FES reaction center, and after passing through the ferridoxine, this electron NADPH produces. This is a total overall of the non-cyclic photophosphorylation. In cyclic photophosphorylation, that is cyclic electron transfer system, the electron is transferred to the ferridoxine to the cytochrome B6F complex. Here Photosystem II does not associated. So This is a non-cyclic and cyclic photophosphorylation. So this is an overview of a complete non-cyclic photophosphorylation. Basically, this is called photophosphorylation because here the process of utilizing light energy from photosynthesis to convert ADP to ATP. Now the question is how ATP is produced. So here the proton moves towards the lumen in two cases. Number one, when H2O is splitted, O2 is produced as a byproduct. Electron jump to the photosystem II and through cytochrome B6F complex to photosystem I. And another one is during the transfer of proton from blastoquinone to cytochrome B6F complex, a Q cycle each occurred, resulting proton was transferred to the lumen region of thylakoid membrane. These all protons are further transferred in stroma via ATP synthesis, producing ATP. So from this proton, ATP is produced. So now comes to the conclusion part. So here we see that complete oxidation of two molecules of H2O, one molecule of oxygen is produced and 2 NADPH is produced. During photophosphorylation, the protons are produced, which was created a proton motive force in the lumen of thylakoid membrane, and ATP is produced by ATPase enzyme, which was also called F1H0 partition. By regulating the partitioning of electrons between NADP plus reduction and cyclic photophosphorylation, a plant adjusts the ratio of ATP to NADPH ratio produced in the light-dependent reaction. This NADPH and ATP, which is utilized in the dark reaction of the photosynthesis, which I discuss in the later class. Thank you. For further class, kindly subscribe my channel and press the bell icon for quicker notification.